So I'll just jump right in because I'm going to try and cover a lot of ground. I've tried to set up the data business in a way that people who might not be as familiar with it can try and follow what I'm trying to say, and it helps me follow it as well. So just if we think about pediatric urology over the last 30 years, I sort of jumped in as a senior fellow with admitting privileges in Seattle back in '94 and look at some of the main themes. Hydronephrosis, for example. The conversation back then was, when and on whom to operate, and we used ultrasounds and scans and manage the bladder with anticholinergics and alpha blockers. And in 2024, looks largely, in fact, the same. Hypospadias, the conversation had been around how to operate and a lot of conversation on technique with a very high complication rate, as you know, for proximal hypospadias. And in 2024 is looking pretty much the same to us. And UTI and reflux had always been a big topic, and prophylactic antibiotics and reflux surgery was very big back then. And again, it's pretty much the same, except that the surgery for reflux has practically disappeared. So that kind of reminds one, or me anyway, about this little joke cartoon about this man who tells a policeman he lost his keys in the parking lot. And the policeman asks, well, then why are you looking under the lamppost? And the man says, and we're not the only people with this issue, but we're studying only what we know and we're measuring only what we can measure. And my own bias is that pediatric urology needs new variables even to begin to ask questions. We need to dive in to understand mechanism of disease. We don't understand the mechanism of disease for any of these four major areas, for example. And I think that's going to be important to drive new questions and generate new hypotheses. So that's why, and that's been the thing that's inspired me over these years, and what I wanted to share a little bit of with you today. I have one disclosure here with the Journal of Frontiers in Urology. So I wanted to try and cover sort of a wide range of things in a short time, but get you to appreciate some smooth muscle biology and the inductive role of the extracellular matrix, which is the collagen and ground substance that surrounds all cells, and understand some epigenetic responses of bladder smooth muscle cells to the matrix, to appreciate some normal cell function and gene expression in hypospadias, and again, give you some early examples of epigenetic rescue of gene problems in hypospadias, and finally, something similar for the role of epigenetics in potentially in UTI. So I just want to say one quick word about AI, which has been something that's hitting medicine like crazy, as you know. And back in 1995, we actually looked for the very first time. I had the idea: could artificial neural networks be used to predict outcomes and help us follow issues related to pyeloplasty? And it actually worked very well. This was a collaboration with Craig Niederberger in Chicago. And now, fast forward to present day, Mandy Rickard and Armando Lorenzo and our group have been incredibly prolific in the last few years in using machine learning and AI to ask questions about hydronephrosis and pyeloplasty and outcomes. But we all acknowledge one critical limitation, that is using surgery as an endpoint in these models, because a surgical decision is never part of the natural history of a disease. And I think it's important to realize that. And we acknowledge that, and I mean, so some very exciting things are coming out of this, but we have no available natural history understanding or biomarkers of what we look at to include in any of these AI models, let alone in any of our clinical predictions. So we've been interested in primarily in bladder outlet obstruction, and of course, posterior urethral valves is our prototype diagnosis, but this applies to BPH and other obstructive phenomenon. And just to remind you, the basic pathology that goes on here is something we call fibroproliferation, a lot of fibrosis that drives a clinician crazy, buildup of smooth muscle hypertrophy, hyperplasia, a lot of extracellular matrix accumulation and derangement. And the bladder smooth muscle cells in these bladders lose differentiation. They lose their ability to contract properly. And this is a feature of cells that are kind of going backwards developmentally and are having a problem. An increase in the type 3 to type 1 collagen ratio. There's a flip, altered blood flow, and obviously changes in growth factors and signaling are some of the fundamental things that are understood. The clinical challenge for us is that despite treatment, and we do TR valves and TURPs and using drugs and that sort of thing, we know that there's there's a persistence of this tissue pathology in some of these patients, despite what we do. Not all bladders become normal again after we treat them, either with drugs or removing the obstruction. And this is true even of bladders that were normal to begin with, and neurogenic bladders after pharmacotherapy. So we are relegated, and have been for decades, to treating symptoms with what you can see here, surgically and medically. So the real question that was interesting to me is, biologically, what are some of the things that might be going on here? So we got interested in the extracellular matrix way back when I was a resident in Boston at Children's. I was working with Mike Freeman, and we started with doing work with this NBMC2 prostate cancer cell line that, Martin, you might remember, was developed in Leland's lab. And it's a non-metastatic prostate cancer cell line. It's relatively well behaved. It grows in these nice defined islands. But when we cultured them on this extracellular matrix gel, which was isolated from a sarcoma, an animal sarcoma, this stuff was available commercially back then, and put the cells on this gel, something very interesting started to happen. And it's important to understand that we use single cell clones here. So there wasn't a mixture of cells we were talking about. These were all genetically the same. And we put them on this gel. It was a 0.5 millimeter thick, which was a lot of thickness for a single cell layer. And the cells moved through this gel. And this is what happened. Within about a couple of weeks or so, they went completely bananas. They transformed their phenotype into a very fibroblastoid, highly migratory, highly invasive cell type. They destroyed the gel. They took on unique gene expression. You can see how we tracked cell motility back then as well. And we repeated this kind of experiment many times. The same thing happened every time. And the really fascinating thing was that this induced aggressive cell phenotype, it persisted even in the absence of any more of the extracellular matrix gel. So you're thinking, whoa, what's going on here? There's no mutation happening in just a couple of weeks, and it's happening every time we do it. So the earlier paradigm that we and many others have been working with about thinking about the extracellular matrix is that injury, in this case obstruction in the bladder, and changes in growth and function of the cells result in the fibrosis that we see clinically in these tissues, and that the matrix is basically a passive sort of bystander, if you will. But a very prominent cancer biologist in Mita Bissell in California in the 70s was promoting this idea that she called dynamic reciprocity, where the matrix itself and some of the surrounding stromal cells, if you're talking about cancer, actually have a powerful influence on the original cell type to begin with. And so that brings up the possibility of a novel way of thinking about this, that the ECM itself regulates bladder smooth muscle cell growth and phenotype. And sort of the bladder smooth muscle cells, even though they're damaged, are making this abnormal matrix, but there's this byplay now between the matrix itself and what the cells are doing. So in one of our earliest experiments, we plated bladder smooth muscle cells on simple sort of vanilla type collagen substrates and plates. And as a comparison, we plated cells just on the plastic, didn't put anything else down. So the cells are forced to manufacture their own matrix, which isn't quite as normal. And we saw a small growth difference here. And we were interested in signaling at the time. And when we blocked one of the principal MAP kinase signaling pathways here, as you can see in blue, we saw a small effect. And it wasn't as exciting as we thought it might be. I'm just going to show you this painting that Lapchi Choi had showed us in a talk he gave. He's a very internationally prominent geneticist who discovered the cystic fibrosis gene in Toronto many years ago. And he showed this to us, and he said, "What do you think the title of this painting is?" And there's a lot of prominent stuff in the picture that you could imagine might be the title of the painting. And it turns out the title of the painting is Two Swallows. And the reason it's important and makes a point here is that sometimes small differences can be really important and should be thought about even if they don't necessarily look exciting initially. So we had almost discounted this result at the beginning. And it turns out when we plated these cells on a much more deranged collagen substrate, which was we just boiled the collagen and it's called heat denatured collagen. It's like putting an egg on a frying pan, the albumin turns white. Same idea. You can see a very powerful proliferative response, a very strong hyperplastic response, a lot of protein synthesis that goes on a little later at 48 hours. On the right, you see that the intensity of the red staining, which is smooth muscle actin, starts to drop. The cells start to lose their differentiated phenotype. And now we see a very powerful response to blocking or MAP kinase in this setting. So this is kind of what launched us into this area of study. When we started to inquire about what's the role of bladder distension, pressure, tension, the things that happen in an obstructed bladder in ECM remodeling, we began to work with whole bladders. You can actually take the whole bladder out of a mouse or out of a rat and culture it. There's something called the Langendorf heart model, where whole hearts are taken out of rodents and cultured and studied. So we thought, well, maybe we could do this with the bladder as well, expanded to define pressures. And when we do this, it was work that JP Capoliccio had done when he was in the lab with us. We saw the interesting inversion of type 1 and type 3 collagen that I mentioned happens in obstructed bladders, and a real increase in collagen 12. Probably never heard of collagen 12. There are 28 different collagens out there. 12 is a small linking collagen that keeps that brings the other longer fibers that you're more familiar with together and makes the matrix even stiffer. So these are the kinds of responses that we saw in this kind of model. In addition, sustained bladder distension activated metalloproteinase activity, MMPs. And for those of you not familiar, MMPs edit the matrix. They cut up collagen into little pieces and expose new areas, and the molecules and the collagen fibrils are responsible for remodeling the matrix. And the black you're seeing in that big square picture in the upper left, is a way of actually staining for MMP activity. I'm not showing you the control, but the control has a lot less black in it. And the interesting thing is, if you take the conditioned media from these bladders floating around in a tube and culture naive bladder smooth muscle cells that are otherwise normal, they respond with a very intense proliferative response. So this MMP activity is soluble; it's released into the medium, and when we use this GM 6001 thing, is simply an inhibitor of MMPs to show that this is one of the molecules that's responsible for the response. So this allowed us to sort of develop a bit of a picture of what's going on, that the extracellular matrix, which is also acts like a sponge and holds on to a lot of growth factors and other important signaling molecules, in the presence of excessive stretch or excessive contraction that happens during obstruction, sets up this cycle of MMP release. Growth factors are released along the lines of the MAP kinase that I mentioned earlier, and despite our symptomatic treatment, keeps this biological process going on as a vicious cycle. So this might be one of the things that keeps some of these organs functioning and looking pathological, despite how we've tried to intervene therapeutically. So what about TUR valves, TERPs, anticholinergics? Why does bladder muscle pathology persist despite some of these maneuvers? What's the mechanism? And so this takes us into an interesting area, and I've called it permanent non-cancer phenopathology. And what I mean here is the following. This was a little experiment that Earl Cheng and his group in Chicago had done, Paediatric Urology in Chicago, where they simply cultured bladder smooth muscle cells from neurogenic bladders. That's all they did. They just took them out, put them into primary culture. And at about a week or so, you see that intrinsically, they already have a higher proliferation rate than normal cells. They have a higher response rate to angiotensin II. In our own denatured collagen model, when we take the cells from denatured collagen and put them back onto normal collagen, you notice that we do not see them return to the growth rate of cells that were put on normal collagen to begin with. So something has happened because of exposure to these abnormal environments, either physiologically or experimentally. And back then, in 2003, Dan Hertz had done this work with us when he was in the lab. We had actually used the word for the first time that some bladder smooth muscle cells may be becoming epigenetically altered through interaction with the ECM. So that brings me to epigenetics, and some of you might be very familiar with this, but I think it's a really important part of biology that is good to understand So I'm going to spend just a couple minutes on that. And what this is a way of regulating gene expression, often by silencing or turning down the activities of genes. It can be the opposite, in the absence of any gene mutation. And it's usually by altering how the DNA is packaged or chemically altered through adding little methyl groups, for those of you who don't have a heart attack thinking about organic chemistry back from college. But if we just zoom in on the chromosome for a second, you may remember that DNA is wrapped around sort of 4 principal histone proteins. And as far as the histones are concerned, when they are chemically altered, usually by acetylation or methylation, there are a number of ways that can happen. When those molecules bind to the histone proteins, the DNA unwinds. And when the DNA unwinds, it's available for transcription and for gene activity. When these chemical groups are removed, the gene activity is generally silenced. You see the nucleosomes coming together, and there are a whole class of enzymes that kind of zoom in and remove these chemical alterations from the histones, and the DNA tightens around the nucleosome and is now not available for transcription. So these are ways, no mutations happening, no change in the DNA sequence, and yet a way that gene expression can be regulated. The other big area in epigenetics is methylation of DNA. You remember A binds to T and cytosine binds to guanine in DNA. And cytosine is the particular site or nucleotide that gets methylated in DNA by a whole class of methylation enzymes known as the DNMTs, the DNA methyltransferases. And DNMTs have a particular predilection for longer runs of C and G in DNA. They're called CPG islands. And these things are what seems to attract the highest degree of methylation. And this prevents the machinery that comes in to start gene transcription. So when DNA is methylated, generally gene transcription is shut off. The opposite can happen, but that's generally how it works. Now, when we started this work, we were pretty much stuck with a relatively straightforward ways of studying this that we use. And one is to just look at the movement of the methyltransferases into the nucleus, because that's where the DNA is. So if suddenly the enzymes are going in there, then something epigenetically is probably going on, as well as their levels in enzyme activity. And we looked at whether particular target genes were increased or decreased in expression, but was that increase or decrease also associated with changes in methylation? That's primarily how we've done things. Things have become far more sophisticated now in the epigenetic world. So a couple of other comments. DNA methylation, these marks they're called, can be inherited across cell division, and they can actually be inherited through generations of people, like from your grandparents to your parents to your children. And they're thought to be important in the stable perpetuation of disease states, which is exactly the sort of ongoing obstruction pathology we were talking about in the bladder. They're also detectable as markers, and this has become a very interesting and hot area of work, certainly in cancer biology. And epigenetics is a principal way that cells can respond epigenetic alterations to their environment, to environmental changes in pressure and stimuli. And when you think about it, a lot of pediatric urology conditions are a response to external stimuli. These things like mechanical obstruction, environmental, hormonal xenoestrogens and things, and certainly infection, these are all far too common to be causing problems in tissues in patients through gene mutations. So to me, that sounded like an attractive reason and rationale for studying epigenetics in the first place. So coming back to this denatured collagen model of ours, one of the first things we looked at was when we inhibit the methyltransferases in this simple growth assay on denatured collagen, we were actually able, for the first time, to suppress the growth response that I showed you earlier, suggesting that there may be, to a certain extent, some epigenetic response mechanism going on here. This is another example you see in the yellow arrows in the bottom left, nuclear translocation of the DNMTs into bladder smooth muscle cells on the denatured matrix versus the normal, and also a concomitant loss of staining of smooth muscle actin as well. So they're losing differentiation again at the same time. When we looked at sort of a higher level, there are various assays one can do now to look at these CG, CPG sites in DNA, they're called, to see where they're methylated. When we looked after only 48 hours of putting normal bladder smooth muscle cells on this abnormal collagen, we saw a 2% overall change in methylation in these 487,000 sites that the assay showed us, which compares actually rather strongly to other models of epigenetic change in other diseases like cardiomyopathy, and in one case, cancer versus normal. 2% change in methylation after only 48 hours is a pretty dramatic response. And some of it was hypermethylation, some of it was hypo. There are various statistical methods one can use to actually look for what are the most prominently altered genes in systems like this. So I'm going to move from there into talking about obstructed bladder modeling. Most studies that look at this topic examine what's going on during the genesis of obstruction. Rodents are generally used, mice and rats. And the thing to remember is that if people doing work in this area, we contend with this all the time. These models are difficult to reproduce. They're difficult to standardize. There's very high mortality. It requires a large number of animals. So we looked at this problem early on. Martin Seiler was a postdoc in our lab, developed an alternate way of obstructing bladders to move to the mid-urethra instead of at the bladder neck, where there was a lot of important neurological things going on at the bladder neck, which created a lot of noise in these animals. And particularly when you're talking about epigenetics, any sort of perturbation just experimentally can create a lot of noise that you may not be interested in. So we called it Finding Nemo, which stood for mid-sparing mid-urethral obstruction. And we did this first in females, and then more recently have been able to bring this model to the male rodent, which is a little more difficult to do, but you can see the suture just below the pubic symphysis here. And it's important for anyone considering studies of any kind in epigenetics to be applying what you're doing and considering what you're doing both in males and females for hormonal milieu reasons, amongst others. And I'll touch on that a little later. So to be able to bring this kind of model to both sexes is very important. This is just to show you that it's possible to look for correlations between changes in gene expression, clinically relevant bladder structure functional changes in animals, and changes in methylation state. To do that, Martin had also come up with a whole series of bladder parameters that one could measure non-invasively using metabolic cages in a way that doesn't catheterize or invasively get into the bladder in these animals. And this allows us to do comparisons like this, where you see changes in gene expression in blue, changes in physiologic parameters in green, the gray box where the intersector are the hottest areas of methylation changes. And this allows us to get, using these large databases that biologists use all the time, to try and understand what are some of the important genes that might be epigenetically altered in this kind of system. Now, another thing that has not been talked about very much is the de-obstructed state. Because to understand what's going on, we need to be able to model what happens after obstruction. The molecular events in the aftermath are terribly important, because by the time we see patients, these bladders have already been obstructed. So it's nice to experimentally ask questions about what's going on during obstruction in animals, but that doesn't really teach us too much, because we get the patient after all this has happened. So how do we model that? And to do that, we came up with something that we ended up calling chronic obstructive bladder disease. We borrowed the term from the respirologist, COPD, and we called it COBD. And basically in this model, after 6 weeks of obstruction in a rodent, 4 to 6 weeks, depending on whether you're dealing with rats or mice, versus 2 as a control, and then 6 further weeks of de-obstruction, we go in a second time surgically and remove the obstruction. On the right, you see that these bladders persist with the pathology that I'm talking about. Consistent smooth muscle hyperplasia, hypertrophy, dedifferentiation of the bladder smooth muscle cells. And you can see from the histological and functional pictures how we've modeled this after obstruction, de-obstruction phase. So this allowed us to begin to work with a hypothesis that epigenetic responses in smooth muscle are determinant in this post-obstructive persistent disease state. And just a few years ago, we showed that if you use a DNMT inhibitor in these animals in this phase of the disease, we actually saw some degree of preserved micturition volume, some recovery of voiding efficiency, and even a reduction in the collagen that was being deposited in the wall of these bladders during this phase. There was also some reduced mTOR signaling, which, as some of you know, is controlled by inhibited by rapamycin. And in previous work, we'd shown rapamycin to actually improve the obstructed bladder state. And in vitro, if we add rapamycin to a DNMT inhibitor, we get even better recovery of the green smooth muscle actin staining. So the idea of combining different approaches epigenetically and non-epigenetically gets put on the table. So as far as therapy is concerned, one needs to think that to cure a disease that's epigenetically based, you need to reverse the epigenetic scar is the term that's used. And DNMT inhibitors and rapamycin are probably too broad to be used in a non-cancer setting. So Karen Aiken, who's been in my lab for almost 30 years that we've been there, I started to work on this proof of principle idea, and we used one of the genes that came up in the screens. It's called KCNB2. It's a potassium channel gene. It doesn't really matter the name, but it was a gene that was downregulated and hypomethylated in this case in this COBD model, and just shows you a gel contraction assay. You plate bladder smooth muscle cells on these little bits of collagen in a gel. On the very top in white, you see the gel's all spread out. It's not contracted. And on the bottom, the positive control is carbachol, which causes them to contract the gel. And when we genetically go in and inhibit, create this artificial downregulation of KCNB2, we also see the gel gets contracted. So the quick takeaway here is that KCNB2 under normal circumstances may support normal contractile behavior. And when it's hypomethylated and downregulated, it might actually be supporting hypercontractile behavior in smooth muscle cells. So then we went a little further using a CRISPR-Cas9 guide RNA system, and this is purposely complicated, but you don't need to worry too much about it. But the idea here is that can you go in and remethylate a gene to its normal methylation state? And basically the guide RNA takes CRISPR, which is tethered to the catalytic domain of either the methylation enzyme or the demethylation enzyme, depending on which way you want to go, and target the specific enzyme at the specific site that's being methylated or hypomethylated in a gene. And it turns out that when you use the guide RNA in this way, you can see a recovery of KCNB2 expression, as well as a recovery of differentiation, calponins, another smooth muscle cell marker in a system like this. So the clinical implications here are that multiple genes may remain epigenetically altered following deobstruction in this deobstruction phase. Multiple guide RNAs could potentially be delivered together. They could be tailored to specific epigenomic signature in a patient's tissue, in the patient's bladder, because they may not all be the same in everybody. And so that opens up the idea of personalized medicine. These kinds of things can be injected into the bladder muscle the same way we inject Botox. And there could be a potential here in later work to look at the possibility of reversing this epigenetic scar and open the opportunity to epigenetic marks and biomarkers in therapy in this disease. So just to quickly summarize this part, bladder obstruction triggers ECM remodeling. Deranged ECM has obviously these profound effects that I've shown you. DNMTs regulate bladder smooth muscle growth and contraction. In valves and other obstructive diseases, this chronic post-obstructive state is probably a very important thing to understand. Epigenetic inhibition can mitigate aspects of that, and there could be the potential to target specific epigenomic signatures in this disease. I'll just move quickly into these other couple of areas, which are far shorter than the first one I showed you. Most of our time has been spent in bladder obstruction. In pediatric urology, when we talk about hypospadias over these past decades, it's been in a surgical setting about how to move the skin around, how to bring vascular tissue in, how to do the urethroplasty, how to fix curvature, whether stents are needed or not. But these kinds of discussions are obviously not going to take us any closer to understanding mechanism of disease here. And an interesting exercise I did back in 2018 in Web of Science, there's a way to actually illustrate what the literature is saying. And these are showing you where the hotspots of keywords in various publications are on this particular topic. And you can see, and it shows linkages and citations, how they're connected together. And there's another area in hypospadias where biology and environment are also published and talked about, and the connections are there as well. But the interesting thing to me, which is not surprising, is that these are two disconnected silos in the literature. Nobody's talking to each other about what's going on, and perhaps more relevant questions that are relevant to us. So clinically relevant mechanism of disease questions, if you will, in hypospadias could be things like, how are cell responses involved in development and wound healing, for example. Simple things like cell migration, proliferation of cells in these tissues. What's the effect of testosterone and estrogen? And do epigenetic mechanisms play a role here, particularly given xenoestrogens out in the environment and the rise and incidence of hypospadias that some of you might be familiar with. So this just shows you that we can actually culture genital tubercles from rodents and study over time-lapse movement of cells in these tissues. And I only put these things here to illustrate the kind of things that can be studied, how cells are moving and intercalating. You see in the bottom left-hand corner, the yellow, blue, and red spots have sort of redistributed themselves in the first hour and a half under normal conditions. But in the presence of estrogen, there's no movement in this kind of endpoint. Looking at simple migration of human cultured foreskin fibroblasts from sort of normal circumcisions in hypospadias patients, we see that migration intrinsically, the ability of cells to move intrinsically, is lower with being stimulated by testosterone in hypospadias, and the same for simple cell proliferation. Obviously, a host of genes are involved in genital tubercle development, and Hox and Wint are a couple that we've looked at. And again, in the presence of, this is done in human cultured fibroblasts from hypospadias and circumcision patients. Estrogen suppresses the expression of some of these important key genes. But interestingly, if we come in with inhibitors of DNMTs, and EZH is another enzyme involved in methylation, we can rescue the expression of these key genes. So this suggests at the very surface that there may be something epigenetically going on here with respect to all the estrogen floating around out there. This was a very interesting result that Karen did. A typical hypospadias model in rodents is to just feed the mothers a lot of estrogen, and a lot of the male pups will come out with hypospadias. So in a model like this, we harvested the male genital tubercles, cultured them intact for several days to wash out the estrogen that were being fed, and look at this. Two key hypospadias genes, fibronectin and Wnt, continue to be suppressed after these mothers were fed estrogen, and there's no more estrogen present in the tissues. Again, further suggesting, and at least beginning to make one excited, there might be some epigenetic mechanisms going on here in hypospadias. And something I call the testosterone paradox. I don't know if paradox was a good word. It might have been irony instead of paradox. When we looked at testosterone response to Wnt and androgen signaling, the DKK1 gene doesn't really matter. That was just the readout we were using. We saw a poorer response to this pathway that you would expect to be important in genital tubercle development in hypospadias fibroblasts. And you remember aromatase transforms testosterone into estrogen. And interestingly, in hypospadias, testosterone activates aromatase more in the hypospadias tissues than it does in the normal. So that's kind of a kick in the head, and makes you think that, I showed you the cell motility and proliferation are abnormal, and without gene mutation, key genital tubercle genes in these male offspring can be permanently perturbed. But this blunted testosterone response and exaggerated aromatase activity may be promoting some kind of local estrogen-rich milieu that might be driving what's going on at the cellular level. And downregulation of key genes may be under epigenetic control. So we haven't done a lot of work in this area, but it certainly suggests that there are opportunities for discovery of markers of causality in environmentally induced hypospadias. Could this aberrant motility and movement and proliferation of cells be tied in some way to predict wound healing, particularly when you think about the massive dichotomy in wound healing between distal hypospadias and proximal hypospadias? And can markers and manipulation actually help with prediction or even prevention of this condition when you consider how common it is moving forward? Finally, a role for urothelial epigenetic responses in urothelium. We're all familiar with the use of prophylactic antibiotics in these kinds of clinical settings. And why is that? Why are these the options? Because there's a big concern for urological dead space and infection, and which patients are at risk, we don't know. So everybody gets prophylaxis. There's no predictive biological test for propensity for UTI. This promotes widespread use of antibiotics and triggers surgery. So really, UTI is this big problem we need to understand better biologically. And so much of what we do in pediatric urology is it's a high stakes game based on making this diagnosis of infection. So a lot of other things followed from this. So we need a better understanding of what's going on. And imagine if we could have a customized approach to managing urinary tract infections. If we could predict in some way recurrence risk on a biological basis, this could result in a whole paradigm shift in who we treat. You're familiar with all kinds of other markers for other diseases. It's almost unfair that we don't have anything similar out there. We could use it to correct high-risk patients, repair things sooner, tailor monitoring. So it becomes immediately apparent if there was something like that. So thinking about this, we advanced this hypothesis that uropathogenic E. coli can induce epigenetic alterations in uroepithelial cells. And we were actually the first to show this in the literature back in 2011. In this particular case, we were looking at downregulation of P16. P16, some of you may remember, is responsible for putting the brakes on overgrowth of cells. But you know that after urinary tract infection, you get quite a bit of excessive urothelial proliferation. Might this be one of the reasons this is happening? This also prompted a commentary at the time. And we used a couple of different bacteria from Scott Hultgren in St. Louis, who's done a lot of molecular biology in infection. And he gave us a pair of E. coli. The FIM negative ones you see in the bottom left-hand corner had a point mutation that prevented the bacteria from producing fimbriae. And as you know, as urology residents, the fimbriae are important in the pathogenesis for E. coli. And so the FIM positive ones in the upper left-hand corner are denser red translocation of DNMT to the nucleus. And you can see the differences here in the FIM positive versus FIM negative. But I just, for a few minutes, want you to just park in the back of your mind the fact that some of these urothelial cells are showing increased DNMT translocation, but there are no green bacteria around them, but they're turning red anyway. So I want you to keep that in mind for a second. Same thing was true when we looked at EZH2, which is another methylation enzyme in the epigenetic machinery. And this just shows you the hypermethylation and downregulation of P16 that we had observed initially. Now, I just finished saying we don't need bacteria. When we use soluble LPS, the toxin that uropathogenic E. coli make, same thing happens. You get this tremendous activation of DNMT in these are a couple of different human urothelial cell lines. And when we use conditioned media from cells that have been infected, just strain out and filter out all the bacteria in the cells, just use the conditioned media, same thing. There's something something soluble here going on after infection that can activate this machinery. And this seemed to be specific to urothelium. When we exposed human foreskin fibroblasts to LPS, we did not see any particular epigenetic activation going on. So this wasn't a sort of a non-specific phenomenon by any means. So how might epigenetic signals be transferred between cells? And can UTI-induced marks be detected in urine, which would be really kind of a cool thing to think about? So we turned to thinking about exosomes, and for those of you unfamiliar, there are a lot of intracellular vesicles that cells make, and exosomes are being studied very intensely in the last 10 years or so as a very powerful cell communication mechanism for moving substances from one cell to another. And they tend to be very rich in epigenetic cargo. that can cause these kinds of responses in other cells. So you can isolate exosomes from any kind of tissue you might be interested in. We've done it from our whole bladder. We've done it from urothelial cells, healthy or post-infection. There are kits that are available to do this. It's not quite so simple, but it's becoming relatively mainstream now. And when we take infected urothelial exosomes from urothelial cells in vitro and expose naive uroepithelial cells to them, you see, again, this activation of epigenetic machinery in the cells, and the green around the far right are these post-infection exosomes glomming onto the urothelial cells themselves. Saw the same thing here with EZH2, but the interesting thing here is that exosomes from healthy cells that were never exposed to infection, when concentrated and placed onto urothelial cells, actually showed us a small downregulation of expression of this particular enzyme. And the interesting question that raises is, can exosomes from urothelium, if collected and concentrated, be actually used in a preventative kind of way? That's the kind of question that comes up from doing these simple experiments. I'm not saying that we've shown that, but these are the kinds of questions that start to come up when you examine these sorts of things. And as far as a biomarker is concerned, we put forward this idea a number of years ago that if one was to imagine progressive soluble, as I showed you, transfer of this activity down through the layers of the urothelium, possibly via exosomes, then obviously if the progenitor cells and the basal cells that reproduce and repopulate the urothelium have epigenetic marks in them, those are going to be inherited up through the rest of the urothelium. And the very top layer could actually be examined in urine, in washings, and one might be able to detect these kinds of marks in shed urothelium. And just as a very last slide to show you, at least in the 3 urine samples we used from 3 research volunteers, one can isolate and detect methylation marks in human urine. And this is being done quite commonly now in many other domains of biology. So the idea that being able to come up with a marker that might be informative and be able to be correlated, say, with UTI recurrence risk, were one to move on to a clinical study like that, becomes a very real possibility. So with that, I'd like to end and thank everyone who's been responsible for this work, particularly Dr. Karen Aiken from my lab, Mark Seidler, Dan Hertz, JP Capoliccio, and others, and our funding agencies. And if there's any time left, I can take a couple of questions, but thank you again very much for the experience.